about hormones. So I know that you've gone through some lab experiments to familiarize yourself with hormones. Now we're going to get into a little bit more of the physiology aspect of it. And here we went through this last week looking at the idea of signal transduction patterns in general. So this is like a big picture idea of how signal transduction happens, no matter what the signal is, pretty much. And so here they're showing how you have some kind of signal that the plant is assessing from the environment or something going on internally in terms of its developmental processes, its life cycle. And it needs to dock into a receptor protein. And that's really what kicks off the idea of getting that signal heard by the plant tissues and cells. And so then once that docking into the receptor has happened, you get the process that goes internal into the cell, or I mean, not all receptors are on the surface of the cell. So we have to think about that too. But basically you have to get essentially that signal transduced, amplified and transduced so that the right genes can get turned on or off, so that you can get a shift in behavior by the plant in order to respond appropriately to whatever signal that was initially. And so after a while, you can imagine there's plenty of scenarios where the plant may have to stop that behavior. Like, okay, we've done enough. We've, created a flower, or we've responded to that pathogen, and now we don't have to keep doing that because it's been resolved or it's done. And so you need a way to attenuate, that's the fancy word, you attenuate the signal. And that can happen through a variety of mechanisms, which we'll mention later. Okay, so we've been through that idea. We've looked at the idea that not all receptors are necessarily in the cell membrane on the surface of the cell. We may see some internal internal receptors that are on other membranes, but basically you still have to get the signal chemical docking into the receptor. And very often it can be a hormone, but sometimes it's something else. So for example, if a pathogen is attacking the plant, it may be some bit or piece of the pathogen's body that ends up docking into the receptor, and that flags the plant that there's a pathogen present. And so we'll talk about that too. But this set of slides, we're more focusing on what about the hormones? How do those interact with the physiology of the plant? We looked at second messengers last week too, that you need to often amplify the signal so that it really gets heard loud and clear, and there's no ambiguity, and the plant can really respond appropriately. So they know for sure calcium is a major player, much more than they thought initially. Calcium is a major player as a second messenger. Even shifts in the pH tell the plant can behave like a second messenger idea. Bits and pieces of their lipids coming from their membranes can also serve a role. That can often happen if they're getting attacked. So if you can imagine like a cute little caterpillar chewing on a leaf, it's going to destroy some of the cell membrane material. Those bits and pieces now coming and docking into receptors tell the plant, eek, I'm getting attacked because now I'm falling into bits and pieces here. And so it's a way for them to assess they're getting attacked. And then we left off, I think, with reactive oxygen species that they kind of serve a dual role for the plant. Like you probably may know for human health, there's, pretty much they're saying reactive oxygen species are bad for you. Well, actually now they're showing some of them are not so bad for you in moderation. And the same for plants. You have this dual role where the reactive oxygen species can be used as a very effective second messenger as part of the signal transduction scheme. And it often is due to some abiotic stress, some non-living thing like drought or flooding or cold. And the reactive oxygen species, if they get to a certain concentration, they will definitely impair growth, impair development, decrease overall yield. But on the other hand, it can serve as a good adaptive trait for them to respond appropriately. So these reactive oxygen species can be used as part of the signal transduction And it can be very valuable information that the plant is getting through these reactive oxygen species. It sounds kind of weird, but it's a dual role. Yes, too much of anything can be bad for you. So sometimes reactive oxygen species are not good for the plant. And we talked about that already with photosystem 1 and photosystem 2 getting broken down by reactive oxygen species. And that's a disaster for the plant. But in this case, we're seeing it plays an important role. They tend, on this figure, they focus very much on abiotic, but we're also going to see when we look at plant pathogen interactions that reactive oxygen species also help alert the plant that they're getting attacked. And so I'm kind of surprised they left that idea off. But anyways, so I put that in there. All right, so let's think about hormones. You've already been introduced to them in lab. So some of this, they are a method for the plant to do long distance communication or between cell communication. And in general, you can say that in plants, the idea that, where these hormones are being produced, it's kind of all over the map. Whereas with human hormones, we tend to have very distinct tissues and glands where this hormone is made in this part of the body and that's it. We don't see that with plants. We see the different hormones are getting produced in different parts, and it depends on the hormone, and we'll get into that a little bit more. They can act right where they're getting synthesized, so they might work right where they get synthesized, or they can work at a distance. As far as I know, with human hormones or animal hormones, those tend to be a distance action. Plants, it can be local or at a distance. They have concentration dependent effects. So we tried to demonstrate that in lab with you when we soaked our leaf discs, the spinach leaf discs, in different concentrations of cytokinin, for example. And we were hoping for kind of an optimum concentration that delayed senescence. Other concentrations either had too little effect or was overboard and caused the actual decline of the tissue because it was just too much. And so concentration dependent. And I give a lot of credit to plant physiologists who do hormone research. My hat goes off to them, so to speak, because it is complicated, very complicated. And so we know that each group of hormones can impact a variety of different events and processes in the plant. And now they also know that hormones can work together on a certain process. And so teasing that all apart and deciphering that all, especially when hormones are made in tiny concentrations in the plant. So just having the technology to pick up on that took a while. And then it's just really complicated. And so I'm not obviously going to get into the nitty-gritty of hormone research because it gets very complicated. But here we have kind of a basic idea of how hormones are involved, kind of looking at this idea of what are they impacting in the signal transduction processes. And here you're seeing, again, maybe it's some kind of environmental signal, maybe cold, maybe, or maybe there's something going on where the plant's like, oh, it's time for me to form flowers. So it's a developmental signal. And then you get hormone synthesis. Often they are transported, but sometimes they can act locally. And then they have to find their appropriate receptor. So again, it's the same idea of where they also have a receptor to dock into. And then that can lead to a signal transduction scheme where ultimately at the level of the DNA, you get shifting in gene expression going on. And then off to your right, you're seeing the idea of a threshold level. So what they mean there is that you can get concentrations that allow that hormone to feed back and start to slow down the whole signal transduction. So again, it's kind of like putting the brake on it. Like, okay, we've done what we need to do. Now we can move on. And so we don't need to keep sending that signal, and it doesn't have to be always on because it's temporary. Okay, so it makes sense that you have this ability to turn off and on this whole signal transduction scheme. So that's what they're trying to show there. And then this other idea of how can you kind of quiet the signal, put the brake on. These are some of the ways. You could pump the hormone out, you could break it down. Catabolism means you're breaking it down. You could hide it away or conjugate it. So we'll learn about that in more detail here. So this is a nice kind of big picture idea to study off of how the hormones are playing a role in the signal transduction process. But also, you know, you do have to have a way to turn it off at some point. It doesn't make sense to always have it up and running. So here are the different groups of hormones that we commonly talk about in an intro phys class. And so I'll go through most of these in telling a little bit about each group. So you've worked with most of these in lab. And then there's a couple of others on here that you didn't get a chance to work with in lab, but we'll still mention them. So typically we start with auxin because it was the first to be discovered. It was actually Charles Darwin and his son Francis. They were avid botanists and they did a lot of very important research on basic plant behavior. And this is one of the books they wrote, The Power of Movement in Plants. He was fascinated with plants that can move, like the Venus flytrap and the sundew, those very dramatic movements, but also just how plants move in general to face the sun, for example. Like, how is that happening? And so he kicked off with his son that research in the 1800s, just doing some very simple experiments to demonstrate that there is some kind of chemical that the plant is making in response to an environmental signal that helps the plant behave appropriately to that signal. And so here on this diagram, you're seeing a couple of different options. So that word auxin actually should be pluralized. I should have done that. So you should put an S there as auxins, okay? Because there's many chemicals that fit into the auxins category. Indole-3 acetic acid is the most abundant that the plant makes, but there's others that the plant can make. And then also, maybe Duncan mentioned this, there's also synthetic versions that we use in research because they're more stable than the plant-produced ones. And so there's plenty of synthetic hormones out there that are just a little bit easier to work with, but they behave the same way as the ones that the plants actually synthesize. So if we go back to this here, one of the ideas of quieting down the hormone and the signal transduction is this reversible conjugation. It says compartmentation slash reversible conjugation. So let's look at that idea. Reversible conjugation. This is actually quite nifty. So plants, you know, if they make their indole-3 acetic acid and it's all good to go, but you need to like, okay, now we need to put the brakes on. Instead of just degrading it and breaking it down, which is effort, and they might need that hormone again pretty quickly. So the option is, well, if I just attach a chemical to it to make it so it won't really dock into its receptor, then it's essentially silenced. And that's often what they do. And that's very elegant. That's a very elegant solution to, we want the hormones available for very quick response, because often the plant has to be pretty quick in responding to whatever signal is coming at them. So they might not have time to make the hormone from scratch. So instead, well, just attach, it's often a sugar or an amino acid that they'll just attach to it. And that pretty much renders it silent. It won't be effective anymore because it can't dock into its receptor. So that's what we mean by conjugation. Okay. So most auxin, when they analyze it, it's actually found in a bound or conjugated form. And then what happens is it's easy to cleave that off, that extra molecule. It's very simple to just get rid of that one molecule, and then the hormone is back up and running. So it's a very nice system and pretty energy. It's a good way to conserve energy and resources. Okay. So Charles Darwin was the first to look at this idea of phototropism. Why are my plants sitting in my windowsill, turning toward the window, turning toward the sun? What's causing them to do that? And so he just did some very simple experiments with his son where they did, did a mock, let's shine unilateral light onto these seedlings. They chose baby seedlings that have coleoptils, which are just special leaves that encase the rest of the baby plant in there. And they're very flexible. And so it's easy to see movement. And that's why they picked them. So anyways, they have that unidirectional light shining, and you can see that if you just keep the plant totally intact, you didn't do anything weird to it, it starts bending toward that unidirectional light. Then they said, okay, what happens if we amputate the tip and do the same experiment? And so that middle picture there, no curvature. It did not respond to unidirectional light. So they're like, there's something in the tip probably that is somehow figuring this out of where the sun is coming from and helping the plant bend. Well, then the critics said, well, but you did major surgery on the plant. That could have been a variable. So they said, okay, we'll just put the intact tip in the dark. And so they capped it with a dark proof like cap and did the same experiment. And they saw, again, no bending. So then they said, okay, based on these sets of experiments, it seems like there's something in the tip of the coleoptile that is being produced in response to that unidirectional light and is somehow causing the bend of the coleoptile toward the light. So after he published that, then a bunch of other scientists got in on this. So here, the next one, Boyce and Jensen in the early 1900s. And you can see they took that set of experiments and came up with some of their own twists on it and started looking at it more carefully. And again, they were showing basically the same idea that there's something going on in the tip. And so they were trying to get at what kind of chemical. And so some of this work that Boyson and Jensen did were showing that it's water-based chemical. It must be dissolved in water. Or it passes through the gelatin, but not through water. Yeah, water impermeable. Sorry, I read that wrong. So yeah, it's a water-based chemical. And so again, they were showing that here, this was a nice one too, where they put a break here with a mica sheet so that if there is a chemical flowing down that side of the stem, the so-called dark side of the stem, the side that's not being lit by light, that it didn't allow for any bending. But if they put the mica sheet on the illuminated side, it did get the bending. So that was a critical experiment where it was saying, okay, we think there's something produced in the tip, and we think that the unidirectional light is telling the plant to go ahead and get that chemical coursing down the so-called dark side of the stem and telling that side of the stem to grow. Grow, 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 grow. And you can imagine if that side of the stem is growing faster than the lit side, that does provide a curvature mechanism. Very simple. And that's exactly what they found with more better technology where they could stain the hormone and show. So in this case, the unidirectional light is coming from the right side here and is striking the plant. And you can see how it's bending, and they tag the auxin chemical. So this is much later. This is after all of the research was done and they were able to verify. So what they're showing is that you have auxin being produced in the shoot tip in exposure to that sunlight. And that auxin is part of the signal transduction scheme to tell the plant, hey, we got unidirectional light. It's coming from the right. We need to start curving to the right to get better access to that sun. And so send auxin, which stimulates growth. Let's send auxin down the dark side and get some asymmetrical growth going so we can bend our body to the light, bend our leaves so our leaves are 90 degrees to that incoming unidirectional light. And that's exactly what they were able to demonstrate. So anytime I talk about going to the dark side, I can't help but think about Darth Vader, because I watched Star Wars over and over and over when my boys were little. And so that's essentially what's happening. You have that unidirectional light stimulating auxin production in the tip. Auxin shifts to the dark side and starts coursing down the dark side of the stem, stimulating all those cells, grow, grow, grow. and then you get that asymmetrical growth and bending result toward the light. Does that make sense? Okay. So really, you can see that this research started in the 1800s and it took until fairly recently before they got this all nailed down. And there's still a lot of active research going on with auxins in particular, because there's such an important group of hormones for the plant. They have yet to find a plant that can survive being an auxin mutant, where they turn off all auxin production. The plant does not survive. It dies very quickly. Auxins are just so important. So far they know that auxins are produced in heavy doses, relatively speaking, in the stem, shoot, stem, apex, so the apical meristem, and then rapidly dividing tissues, which you can imagine, depending on the phase the plants is in, that could be all over the body. It could be some root tissue, it could be leaf tissue, flower tissue. So anywhere you have really active growth going, you can find auxin being synthesized there. So far, this is the only hormone that they're aware of that does this idea of polar transport. It seems unique to auxin. And basically what that means is when you see it being synthesized in the shoot apex, it courses down the stem from apex to basal. That means from top to bottom of the stem, okay? So even if you take that plant and you hang it upside down, it still is going to course from shoot tip to basal, even though you've just reversed the direction of the plant. And that's what they mean by polar transport. It's one way. And so they don't see auxin being produced in an actively growing portion of the stem and coursing up toward the tip. They don't really see that. And so if it's doing polar transport mechanism of movement, I'll show you a picture of this, but they say it's a transmembrane route of getting the hormone from cell to cell to cell on down the stem tissue, which is weird. It requires energy, which is also weird. Okay, so this is a little bit more effort for the plant to do this, which raises some weird, interesting questions. And so we see that there's dedicated proteins that will allow for movement of the hormone through the cell membrane, because it can't just fit through the cell membrane by itself. It needs help. So it has special carrier proteins involved. And like I said, it's independent of gravity. If you hang that plant upside down, it's still going to move the hormone from the shoot tip and move up the stem, because it's upside down, to the base. Okay, so it can do that, but thankfully it can also be transported just through the phloem, which is much simpler. That's a much more direct, simple, there's no membranes to cross, it just whoosh courses through the phloem. So it can do both. And that again begs some questions there, like, why is it doing that? So that doesn't seem to require any energy. It just is a passive, nice flow from point A to point B. So auxins are a little weird in that regard. We also know that microbes that coexist with plants long-term that are termed, well, a lot of beneficial, let's just say this, a lot of beneficial bacteria and fungi can make auxins themselves and excrete that out to the plant. So here's, a microbe making a plant hormone, which is weird. But when you think about it, it's the microbes that have coexisted with that plant. And often it's a long-term coexistence where maybe they were even transmitted in the seed to the next generation, those microbes. Okay. And so beneficial microbes are known to do this, and it helps the plant. It stimulates the plant. Yes, any? Yeah. It doesn't seem like the bacteria or fungi use it. It's like it gives it to the plant.
Like, is that the kind of thing that like generated the auxins that it doesn't?
Yeah, especially like with the nodule formation, I wouldn't doubt that there's some. I don't know if they've looked for it, but they must have looked for it by now, but I wouldn't doubt that the bacteria that do nitrogen fixation are secreting auxin to like stimulate a nodule for me. I want a home, give me a house. unfortunately, there's also some pathogens. There's a famous bacteria that also can produce auxins and use it for evil purposes, to attack the plant. So it's not just the beneficials. So I think I'll mention that at some point, but not right now. We'll get into that a little bit later. This is just the idea of polar transport, where if you take the plant and turn it upside down, you still have flow from the shoot apex down to the base of the stem. And so that's why if you ever clone like Sansevieria plant, the snake plant, you have to, if you take sections of the leaf and put it into the soil, you have to know which section is closest to the apex and which is closest to the bottom of the stem. If you put it in upside down, you'll get roots forming out of the top. So that's confused people in the past, like, why is my section of Sansevieria leaf forming roots on the top? It's because you accidentally inverted it. So interesting phenomenon. Okay, so I'll get a little bit more into action of the hormones after I introduce them a little bit more. So here is another group you worked with in lab, the cytokinins, and zeatin and kinetin are very commonly worked with in the plant. And so here, the zeatin is really what the plant makes. So we don't work with zeatin. Zeatin, we tend to use synthetic versions like benzolamine. I think you use benzolaminopirin when you soaked your spinach leaf discs in the different solutions of cytokinin. But the plant makes the zeatin version of cytokinin, and also bacteria can make that again. And so both good and not so good bacteria that hang out with the plant. Fungi can make it too, even some insects. So there are some insects that, if you've ever seen like galls on stem or branch tissue, some galls, they look like tumors. They're actually the result of an insect stinging the branch tissue over and over and over. And in the venom or the chemical that comes out of the stinger, there's actually some zeatin hormone, and it stimulates the plant to make like a tumor-like growth. And that's where they lay their eggs. The insects lay their eggs. They're young. So interesting interaction there. So benzolaminopirin or kinetin, or it's a little bit more stable. So we tend to work with those in research. Just like what we saw with the auxin picture where you can conjugate it, cytokinins are known to be conjugated typically with a sugar. And then once you cleave that sugar off, you get the active form of cytokinin. So again, a very nice way to quiet it down, just attach a little sugar to it and then it's all quiet. And then when you need it to be active, just cleave that off in one little fell swoop, no big deal. So you already learned in lab that if you soak your spinach discs in the right concentration of cytokinin, it slows down their aging process and keeps them fresh. Here is a pretty good visual looking at the one on the left was modified to have plenty of cytokinin still coursing through its leaf tissue, whereas the one on the right was allowed to go through natural senescence. So right now, plant trees or trees here are going through senescence. It's a natural process to get rid of their leaves. They don't want to hang on to those through the winter. And so those leaves are senescing, definitely. And even though we see all those beautiful, beautiful colors, most of those pigments were already there. You just didn't see them because chlorophyll dominated. And now that chlorophyll is getting drained faster than the other pigments. You see the other pigments show up. So everything's kind of getting sent down right now. The trees around here, they're sending all the goodies down to their roots and sometimes into their stems for storage for next spring. We also know that both auxins and cytokinins are heavily involved in the tissue culture. I looked at your tissue cultured vessels just Friday. Unfortunately, a lot of them did get contaminated with that white fungus. I don't know if you saw that. Some of you did have beautiful looking tissue culture still, but quite a few vessels got contaminated. And I believe that was an endophyte fungus because when I checked your vessels at 48 hours after you did your experiment, there was no contamination. And so usually if you see contaminants showing up after 48 hours, it's usually an endophyte that is growing out of the tissue onto the agar, the tissue culture medium. But anyways, the ratio of auxin to cytokinin is pretty important when you do tissue culture. And it really does depend on the plant species you're working with, what that ratio is. But basically, you do have to provide auxin and cytokinin in order for those plants to develop appropriately. So this visual is just showing different groupings of different ratios of auxin to cytokinin. And you can see differences in their stimulatory properties. So here you don't see much of anything going on the top left. But as you work your way, you can see some good callus growth happening here. And in the lower left, you can see not only callus growth, but also you're starting to get little plantlets. You're starting to get root formation, shoot formation, a little bit of leaves going. And so if you hit the right ratio of auxins to cytokinins, you can get that plant to be fully cloned with an intact, roots and shoots and leaves. So auxins and cytokinins are always used in tissue culture processes. Okay, another group that we need to talk about and introduce, gibberellins, again with an S because there's many. And again, fungi are kind of famous for producing this as well. So it was actually first discovered in a fungus, which is weird. And this is the name of the fungus, Gibberella fujikori, and it's a plant pathogen. So it was showing up first they noticed rice plants in the rice paddies were exhibiting, they named the disease foolish seedling disease, because what they saw happening is the rice stems would grow way too quick and too tall and too weak. And then once they started forming the rice grains, the rice seeds, they would start to droop and fall over. Well, that's very hard to harvest rice. And then plus now all the grains are in the water and just perfect for fungi to come along too and start breaking down and using all that yummy food for themselves. So foolish seedling disease. They went into those fields and they're like, what is going on? They discovered big populations of this fungus. And when they studied the fungus, they're like, oh, it's producing this chemical that's telling the plant stem, grow, 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 grow. And that's what's causing the overgrowth of the stem, too fast, too weak. And then they flop over like that. And they discovered it was gibberellic acid that the fungus was making and that the plant was getting overexposed to that. And that was what was causing the plant to fall over. But the plant naturally makes it too, for good things. Okay, so they can make it in their germinating seeds. The gibberellic acid stimulates that development of a good baby seedling. The developing of the leaves to get the leaves fully formed and mature. Stem tissue for sure. It does definitely help increase stem length. So this was kind of interesting that first they found it in the fungus and then they realized, oh, the plant can make it too. And they now know that auxins can stimulate gibberellic acid synthesis. So here again, we see this interaction. And gibberellic acid can also do the reverse. It can stimulate auxin synthesis. So this is, again, really challenging. How do you figure out what behavior is due to the gibberellic acid, or is it due to the auxin? Or so it can get kind of messy pretty quickly here. So gibberellins, if you're curious, that's what the structure looks like. And there's different, for some reason, they've chosen to number these instead of give them unique names. So you see them GA with some numbers, subscript for the different forms of gibberellins. And so after they figured out that plants make this and not just pathogenic fungi, they started playing around with it. And here are some classic images of what it can do. So on figure B and C, you're seeing that they, like the left plant didn't get sprayed. So you did this with pea plants, right? You had dwarf pea and wild type pea. And then the next three plants they did spray. It looks like corn that they did this one with. They must have had a dwarf corn variety. Here again, you can see the dwarf variety underneath the ladder, and then they sprayed with gibberellic acid. And so, and then this figure A is actually, those are grapes on a vine. And I didn't see this until a student said, hey, I think that picture's false. I think the guy's just holding the one in the left closer to the camera. And once the student said that, I'm like, it does kind of look like that, but it's not. And so these are two bunches of grapes, and the one on the left did not get sprayed. Now, they weren't really spraying the grapefruit. What they were doing is spraying the vine. And when the grapes are really, really young and you spray the vine tissue, it causes stem elongation. So it spaces out where the grapes are located. And so they have more physical room to get bigger. And then they usually get much sweeter as well. And so that's been a big boon to the grape growing industry to have that capability. But basically, when you ran that experiment in lab where you had dwarf pea versus wild type, what you were playing around with is that the dwarf plants, they have just one little change in the DNA that codes for the enzyme that does this step of taking GA20 to GA1, which is the very active form of gibberellic acid, the GA1. And so the enzyme, the hydroxylase enzyme in the dwarf, just has a minor mutant change. And that causes them to stay dwarf. They just don't have a very active enzyme under that condition with that one little base change. So they still have the receptor for gibberellic acid. That's why you still got them to shoot up in height, because the receptor was still there. That wasn't the problem. It was just they themselves could not make enough gibberellic acid to grow very tall in their stem tissue. So that was a neat little find that allows us to nicely demonstrate this idea. Okay, so gibberellins, so auxins, cytokinins, gibberellins. And then another group, the ethylene, well, it's just ethylene. And that was the first hormone to be discovered that's gaseous. And luckily, we found a synthetic way to expose plants to ethylene without using ethylene gas, because that's a pain to work with. So there's a different chemical that we can use instead called ethithon, and it's a liquid. And you just put it into the water that you irrigate your plant with, and they'll take that ethithon and they'll quickly convert it to ethylene. So it's nice and handy. But anyways, the plant itself makes ethylene. And we also know that fungi and bacteria that hang out with plants can do this too. And ethylene is a very important hormone for the plant. And I'll get into, like how these hormones work, what they do for the plant in a little bit. But ethylene is very important in a stress response behavior. Lots of different stresses will trigger ethylene, and then ethylene gets involved in a signal cascade. So here's the big Yang cycle for ethylene production. I don't expect you to memorize this. There's a couple key things though I want you to know. So as you come off the Yang cycle, you have S-adenosyl methionine, 8-O-met is the short for that. And then you have this enzyme, ACC synthase, because it synthesizes ACC. You don't have to know the full name, it's just ACC. So 8-O-met goes to ACC with this ACC synthase enzyme, and then this other enzyme, ACC oxidase, converts ACC to ethylene. So that last little side branch, those couple of steps, I would like you to be familiar with, because I'll be talking about this in terms of how we manipulate that in order to, sometimes we don't want ethylene going at full tilt in the plant for human purposes. And so we need to manipulate that to slow it down. And so these boxes that you see, they're trying to show that if you mess around with some of these and the action of these enzymes, you can manipulate like ripening, their response to different abiotic stresses. So we'll get into that. I'll explain more about that later. So basically what I want you to zoom in on is that last step. Okay, so this part of the Yang cycle is where I want you to think about, because we need to think about those two enzymes when we start looking at the action of ethylene. So again, ethylene is promoted by auxins and cytokinins. And so again, if you have auxins and cytokinins, often you'll see ethylene ramping up as well. So then you have three hormones coming into play. And I'll get into this more when we get into some of these abiotic pressures on the plant. We will look at how plants respond to abiotic later in the semester. But basically, ethylene should have been named the stress hormone. Unfortunately, a different hormone got that nickname. Really should be ethylene though, because pretty much any abiotic stress, they do see ethylene getting kicked into high gear and helping the plant deal with that. So you did that with this figure B that you're seeing. You did that in lab with tomatoes when we put the cut apple. So that cutting of the apple and then we put it in the pot and covered it with a plastic bag and then let it sit for a while. As soon as you cut that apple, you wounded the apple. That apple is living tissue. So when you wound a plant, or if a bunny rabbit comes along and wounds that plant, you get ethylene production. And in this case, it causes that weird epinastic response by the plant where it does that asymmetrical growth pattern. So it's kind of weird because flooding can also cause an epinastic response. So you might go out to your field if you're a grower and you're like, we had a downpour, an absolute downpour for days. Why are my plants looking like they're wilting? Because often when you see a plant drooping, you think wilt. And it's not wilting. It's just the opposite. It's the soil is so flooded and so full of water that it's getting denied oxygen, and that can also kick in the epinastic response. So sometimes a wilty looking plant is not wilting. It's actually just the opposite. So you have to be careful about that. So anyways, we'll get more into that later. But definitely ethylene is also very involved in fruit ripening. And we use this knowledge to make sure that fruits are ripened to the right stage when they get put out at the grocery stores for, to get sold. People want, of course, nice fruit, not overripe and starting to decay. And talk a little bit more about that. This image is showing root hair formation. Very, very important. Plants need nice, healthy root hairs. Root hairs are often destroyed as they move through the soil. They're so delicate that you need like constant more synthesis of root hairs as the roots are moving through the soil. Here's a lovely pineapple field in production. You can see all the pineapples there growing. It looks so cute. So we know that ethylene can also delay flowering. So if you are a grower and you want all your pineapple plants to flower at the same time, you might hit certain sections, like if they're starting to flower out of sync with each other, you might want to use ethylene to even it out, so to speak, delay the flowering. But with pineapple, what you do, so this is with a lot of other flowering plants. With pineapple, what you do is if you want to get the pineapple plant to harvest at a certain stage, like you hire the labor, you want things ready to go on a certain date. So you got to get all your timing just right so that you can harvest your pineapple and make your profit margin. Then you hit pineapple plants and mango and a couple others. You can hit it with ethylene at the right time and it syncs up everything. and it actually triggers flowering. In most other plants, though, it suppresses flowering. So basically what we're saying is you can play around with when you want your plants to flower by manipulating how much ethylene they're getting exposed to. It's quite an interesting phenomenon. Here, you might have seen this in the lab lecture PowerPoints, where with this idea of the leaves senescing, what you have happening is, during the summer, you have plenty of oxygen in the leaf tissue. And that's the signal to the plant. I'm healthy, I'm active, I'm doing photosynthesis, everything's great. We're cooking along. And what happens is the oxygen is constantly flowing in low doses down the petiole. And that's telling this area of the plant, yeah, everything's good. Don't get rid of me. Don't cut me off off the plant. But as fall is starting to form or come about, this little symbol here is trying to show that oxygen is going down and it's not flowing down the leaf anymore to the petiole. Well, that is now allowing ethylene to ramp up. So that's what they're showing. And ethylene is being produced in the petiole, which then causes an abscission zone, which is a weakening in a line of cells right there where the petiole attaches to the stem. And if you get a nice weakening, eventually that leaf will fall off. And so they know that it's a process of oxygen levels going down, ethylene coming up, and that causes the abscission zone. So again, these hormones, it's often more than one hormone needs to be heard, so to speak, or needs to be part of a signal transduction in order to have a certain behavior. Abscisic acid, pretty much found everywhere. There's no easy definition of where it's produced. It's pretty much everywhere. And it is, they often define it this way. It's synthesized in cells that contain chloroplasts or amyloplasts. That pretty much means every single cell that can do this. So abscisic acid, again, is just one chemical. It's abscisic acid, which you're seeing here. And we talked about it already a little bit when we mentioned that the idea of how stomata function, the guard cells. And so if you are a plant and your roots are in the soil and your roots are not finding water and you're going under drought, then so that's the part of your body that first figures out, uh-oh, we don't have enough water. And so it's actually in the roots that abscisic acid gets made because that's where it first senses drought. And then abscisic acid quickly gets made. The concentration shoots up very high, starts to flow up the plant body through the xylem to the leaves, to the stomata, to the guard cells, and tells the guard cells, uh-oh, we need to close down. All right. And so it's quite the event because you have to get the abscisic acid that's in the root all the way to the stomata. But it's effective. It kind of makes sense that since it's the roots that have to, experience that drought first, that the warning is there and gets sent up to the leaves. Okay. Okay, so we're going to continue on with the hormones and think a little bit more about how they impact the plant in more detail. So we'll start that up on Wednesday.